Hi, my name is Joshua Guilbert. I'm the Road Office Field Supervisor and the Wildlife Biologist for the Fish and Wildlife Department, currently working on both bat species here in the CMI. This talk is co-written by Emily Kohler from the Saipan Office, Robert Orler and James Mendiola from the Rhoda Office, and Leroy Sablan from the Saipan Office. The Mariana Islands has two remaining bat species. Phenigranus isn't well known, otherwise called the Polynesian sheath-tailed bat. We have our own unique subspecies here in the Mariana Islands, and the Finihi, which is well known, the Mariana fruit bat. We've lost our third species, the Guam flying fox, which was also known as the Finihi, sometime in the 1960s, most likely due to the brown tree snake. But this one was limited, as far as we know, just to Guam. Phenigranus had a historic distribution of all of the southern Mariana Islands and possibly some of the northern Mariana Islands. There's reports from Inatahan, but no confirmation. It may not have been very well established on Tinian. It's a very small bat, ranging from 5 to 7 grams, insectivorous, eating small insects like moths and mosquitoes, living in caves. So it's kind of cryptic, which is no wonder most people don't know about it. Its main threats are the brown tree snake. It's got human conflicts in terms of disturbance of the roost, people entering into the caves. But historically, the biggest thing has been through landscape changes and also the use of pesticides. Typhoons may have also played a role in its reduction and distribution. It only has one pup a year, so it's slow breeding, and its class is endangered by both UN Red List and Fish and Wildlife Service. The exact causes for the reduction of this bat in not just the Mariana Islands, but throughout the Pacific is hotly contested. For Rhoda, it's most likely due to something like DDT, considering the guano piles and the caves that we know that used to occupy are very substantial, so the populations would have been quite large. People saw them as recently as the 1960s, and there wasn't really enough damage or typhoons to account. The one fact that we can confirm is that there was DDT used on the island, so I'm putting my money on that. Currently, they're only found on Agiguan. There are quite a few crevices on Agiguan, indicated by all the little triangles you can see, and some very large caves as well. Previous researchers from various groups have gone out and counted the bats at various stages. Most of the low counts you can see on the graph to your right only were a few bats, and most of them missed the big colony, which is at Black Noddy Cave. The ones that did count Black Noddy Cave I've indicated as the diamonds. Even when they've only count Black Noddy and only a couple of other caves, they get higher numbers than the ones that would count 5 or 6 caves. So Black Noddy is the best place to go to work out what the population is doing on Agiguan. So that's what we did. We've had one trip out so far. This was a scouting mission to try to work out where all the caves are. It was cancelled a little earlier because of poor weather. We had to get off the island till you're getting COVID shots. But we did manage to do 2 counts. We were looking at swiftlets and sheath-tailed bats to see what they preferred in terms of cave usage, to try to work out what sort of caves they'd want to use on Rhoda. We found quite a few swiftlets and only a handful of sheath-tailed bats. But we did make it down to Black Noddy Cave, so we can go back there and count that later in the year. And we located bats flying near Cliff Cave, which you can see here on your right, but we couldn't get up high enough to see whether they're actually entering and exiting the cave. We deployed data loggers. Currently, we have a combination of temperature and humidity loggers. And this is the sort of data that we're going to be hopefully pulling out of it. We'll be comparing the temperature and humidity at the front, middle, and back of the caves on both Saipan and Rhoda to try to work out which caves are the best ones to release the bats into. We're also doing a diet study that Leroy is going to talk about in later session, where we're looking at what the bats are eating, analysing their poo, their guano on the cave floor, and using both genetics and morphological methods. We also hope this year to do some genetic sampling, but because there has been a recent typhoon and hasn't been any recent counts, this is kind of being put on the back burner. But when we do eventually get the permission to do it, we'll be using this thing called a harp trap. You can see me setting it up here in the forests of New Zealand. It's the best thing to catch microbats in. So essentially it's an aluminium frame with taut fishing line. You then put it right outside the cave or the roost entrance if it's in a tree. The bat leaps out, doesn't know that this catch bag is there, and then they just go to sleep. You can then pick them out of the bag, measure them, and take a wing biopsy. This is done by using this little tool with a sharp tip on the end, and to get a little cookie cutter cut out of the wing membrane, which heals over within a few days to weeks. It's one of the bat wing membranes are one of the fastest healing materials in nature. So it's the best place to get high quality DNA. But again, because we don't know what the population numbers are, this has been put on the back burner, but we will try to get some DNA to work out the genetic diversity using bat guano. We're also expanding in FY22 to do getting an environmental assessment done to fulfill our compliance requirements. So if you know of anyone with a environmental consulting company, get in touch with Steve Mullen. We're also going to repeat counts on a Agiguan in April and August and FY22, and then expand to do acoustic surveys on Tinian to see if they've colonized Tinian. And of course, we're continuing our guano collection to collect genetic samples. Most of my work in the Mariana Islands has been done on the Whanihi. This is distributed currently throughout the Mariana Archipelago, although we suspect there's probably very few, if any, bats on Tinian. They are relatively large, weighing up to a pound. They feed on fruit and nectar and roost in trees. They have all the same threats, including the extra human conflict of hunting. They only pup once per year, and they're currently classed as endangered by the IUNC Red List and threatened by the Fish and Wildlife Service. So DFW's goal is to delist the species, and to do that we're using the roadmap put out by the Draft Recovery Plan. To delist the species, we have to demonstrate that they're stable to increasing on 3 of the 5 southern islands. They're doing this on Guam, mainly because bats from Rota are flying over whenever there's a typhoon or a drought, and this establishing there. So the bat numbers on Guam are increasing. The bat numbers on Rota, as you'll see in a little bit, are stable to increasing, and we think they are on Agiguan too. So we may have met that requirement for the southern islands. We do not know what's happening that well on the northern islands because it's kind of difficult to get to there. So we're going to be working towards doing a survey once every 5 years in our 5-year plan. We will go up with our partners and try to actually do a blitz where we sample each of the islands. But again, it's expensive and it's logistically difficult, so that probably will only happen once every 5 years. The same monitoring that we'd be using to delist the species, we would implement as a post delisting to meet requirement 2. Requirement 3 for habitat loss and degradation. I think we've got a pretty good handle on that with the one-start and permitting process in the Mariana Islands, Northern Mariana Islands. The big thing though is still hunting. We don't know how bad that is. We've got a project that I'll explain in a little bit to get more data on that. Brown tree snakes are stopped quite well by the BTS program run out of the Department of Fish and Wildlife. And while there are operations on Tinian and potentially on bombing of FDM and maybe Pagan, military development is not impacting the Mariana fruit bat as much as the hunting or the brown tree snakes will be. So on Rota, previously I showed data up until FY18. We've got 2 more years worth of data since then. The big thing, and each time that has a negative impact on the population numbers, are typhoons. You can see them indicated here by this little blue one for dolphin and red for mancut. But this extended drought period we had also had a negative impact. And when that happens, it drops down the population on Rota down to around 2,000. So I think that's the carrying capacity for the island after a typhoon. But it has sprung back both times, and our data so far for FY21 puts it somewhere in the region of 3,000 to 4,000, but I'm not confident in those numbers yet. We still have a few more months to count. More recently, we have had, during this current dry season, a report of 166 bats suddenly turning up on Guam. So again, our bats from Rota are flying out to those other islands whenever the conditions get tough. So I think if we really want to save this species, we have to make sure that when they leave Rota, they want to stay on those new islands and they can colonize those locations. I think typhoons have probably been a major push for in droughts to the colonization of the species up and down the Mariana Archipelago. So for FY22, we're going to be doing Saipan and Tinian island-wide counts to try to increase our monitoring. There's going to be using members of the public, getting up early in the morning and going to stations to see if they can see bats flying around and then recording the distance and direction. This is similar to the one that they do on Guam. We're going to be doing regular counts on a Giguan in tandem with our Whinihi Garnis program. We've got interviews with hunters coming up. So basically we're signing an agreement with the University of Iowa. We're using their web-based system to ask anonymous questions for both hunters and non-hunters alike to find out what the population thinks the trends are, get data on the hunting pressure, and also feedback on how we should be monitoring the population. And finally, we're going to be doing sound monitoring where we put recorders in the forest to record gunshots and work out which ones are due to hunting of deer and which ones are due to hunting of whinihi based on their location and time of day. If you want to help with any of those, you can drop my email. A special thanks to everybody here and from the Rota office and also the University of Washington that helps out with the rehab program we have for the whinihi, and a great welcome to our new staff member, Daisha, who we stole from them, who's now started working in the Rota office for the Bridled White-eye. Any questions?